Oh, Federica is here.
Oh, perfect.
Yeah,
So, welcome everyone. Welcome especially to Elisa, who is today's speaker. She is a PhD student here in Queen Mary, University of London, and she's gonna talk about some amazing stuff. Elisa, let's try to share the screen. So she's gonna talk about some investigations that she did with mathematical modeling, looking at evolutionary parts of ecDNA, but wait also for the next part. It's going to be a great discussion afterwards about some things, not spoilers. So Elisa, the floor is yours. Go ahead. Thank you again.
Okay, thank you, Gabriele. So first, can you see the screen? Can you hear me?
Yes,
Can you
I
see
can.
here?
Yes, also.
Okay, fine.
Wait.
Perfect. So thank you for inviting me, Gabriele. So today I will try to introduce you to the fantastic world of mathematical biology. I will just introduce you a bit about my project, that is an investigation through a mathematical model of the evolutionary path of a genetic mutation that is called extra-chromosomal DNA. Before starting though, I would like to introduce you, yes. I would like to introduce you to my team, that is basically a worldwide team and a multidisciplinary one. It is called eDynamic, and it's basically a team of clinicians, experimentalists, and theoreticians. And all of us, we are just working on this mutation that I talked to you about before, that is this mutation extra-chromosomal DNA. And our goal is basically understanding the connection between extra-chromosomal DNA and cancer, because there are many evidences that I will show you later about how this mutation is related to the initiation and also to the spread and evolution of cancer. So you can see that this group, my group, is spread in US. In US, we have some clinicians and experimentalists, and they are doing some experiments. They are collecting data, etc. Also, there are some clinicians in Germany, in Berlin, actually. And in London, we are the theoreticians. So basically, we take studies and data from our colleagues, and we try to elaborate them using mathematical models. And we are, some of us are in Queen Mary, some of us are at UCL. But basically, yeah, the theoretician for the eDynamic team, we are just in UK for now. So in order to make you understand what I am talking about, I would like to talk to you about cancer. So I think that every one of us knows what cancer is. Cancer is basically a mass of cells that are growing uncontrollably, and they are basically affecting the functions of our body. At the very beginning, though, cancer is not this big mass of cells, but it is just a bunch of cells that could be easily attacked by the immune system of our body. So here in this picture, you can see that if the size of tumor cells is pretty small, it is possible for the T cells, that are the cells of our immune system, to attack the cancer cells. So basically, cancer, in order to evolve successfully, it has to develop some techniques to escape the immunitary response of the body and to basically grow uncontrollably. And the technique that the cancer cells use to do that is DNA damages. Basically, what is happening is that cancer cells storage the oncogene information, so the genes that are carrying cancer properties, they are storing these genes outside the chromosomes. Basically, so when the cell is dividing, the chromosomes are not carrying oncogenes. So they are basically chromosomes as in the normal cells. So the T cells, the immune system of our body is not able to recognize these cells. But actually, in some structures that are circular and they are outside the chromosomes, there are oncogenes. And they are basically growing uncontrollably without being attacked by our immune system. And these circular structures are actually called extra-chromosomal DNA. And it's the object of my study, basically. Also, you can see here in this picture, we have chromosomes and this small circular structure in the green box. These are extra-chromosomal DNA. So what's going on is that the cancer could escape our immune system and it can grow uncontrollably. thanks to these genetic mutations. So from these slides, you can see how important the study of extra-chromosomal DNA is in terms of understanding how to control cancer and how to also develop new strategies of therapy. Let's talk about some studies and the medical literature that are supporting our ideas. First, ecDNA promotes tumorigenesis. So ecDNA promotes the initiation of cancer. Here on the left, you can see a study that has been done in 2020. There are different tumors here, and some samples of these tumors have been studied. And basically, the percentage of cells that are carrying extra-chromosomal DNA has been calculated. And you can see from this histogram that more than half of all these tumors is carrying actually cells with ecDNA. So ecDNA is definitely present in many types of tumor, and it is massively present in many types of tumor. It is not just that, because also extra-chromosomal DNA leads to a significantly shorter survival for patients. Here on the right, you can see that we are calculating the survival probability over time of a patient with a tumor. And if the patient has a tumor that is carrying extra-chromosomal DNA, then it is in the trend, in the color trend. It is not, it is in the gray trend. And we can see that cells that are carrying the mutation leads to a shorter survival probability. Also, in a more biological view, we can say that extra-chromosomal DNA promotes oncogene amplification. Just to let you know, oncogene amplification is a process where oncogenes, so the genes that are expressing tumor properties, they are growing in a higher number rather than normal genes. And this, of course, because extra-chromosomal DNA is carrying a lot of oncogenes. And as extra-chromosomal DNA is increasing faster than normal DNA, we are expecting that a lot of the cells with extra-chromosomal DNA have a higher chance to express oncogenes rather than normal cells. Here on the left, there is a study, again, made in 2020, and you can see that on the x-axis, we have some oncogenes. And in the y-axis, basically, we have the percentage of samples of extra-chromosomal DNA that is carrying the specific oncogene. And we can see that extra-chromosomal DNA is basically carrying most of all these oncogenes. So it is reasonable to think that extra-chromosomal DNA is highly related to cancer initiation and progress. In terms of creating a mathematical model, for us, it was important to understand how ecDNA is segregated into daughter cells. And this process is pretty nice and cool because actually it is completely different from the chromosomes, because the DNA in chromosomes is segregated linearly. So we have the mother cell with its own DNA. During the cell division, the DNA is doubled, and each daughter is inheriting one copy of the DNA. For extra-chromosomal DNA, this is not happening. So if the mother cell has one ecDNA copy, then into this daughter cell, 2 copies have to be distributed. And they are distributed completely random. So it is possible to have 2 scenarios, for example, 0 copies plus 2 copies, or 1 plus 1. If we have to distribute 2 copies, if we have to let a cell with 2 copies divide, so we have to distribute 4 copies, we could have 1 plus 3, 0 plus 4, 2 plus 2. So we could have all the different combinations. And this is very important in terms of creating a mathematical model, because actually the stochasticity of a ecDNA proliferation path allowed us to create a model with stochastic equations. Also, like the last slide about introduction, I will present you a multiple species ecDNA evolutionary process. So I will present you a model where we have 2 different types of ecDNA. Of course, the question is, am I allowed to consider 2 types, more types of ecDNA? And the answer is yes, because the properties and the fitness of ecDNA is deeply related to some factors. First, epigenetic states. Basically, epigenetic states are external states, external environmental factors that are affecting genes. For example, we know that smoking is affecting the genes that are related to lung cancer. So, for example, this could affect also the properties of a molecule of ecDNA. Carried oncogenes, different ecDNAs could carry different oncogenes. So we can consider different types of ecDNA due to the different oncogenes they are carrying. Third, sites of growth, because for example, if we let a cell culture to grow in vivo or in vitro, the results are completely different, especially for the reproduction rates of cells that are carrying this mutation. And finally, stress condition, because hypoxia or high acidity of the body, they are an advantageous environment for extra-chromosomal DNA cells to grow faster. Okay, after all this introduction, that was just for you to understand what I am talking about with my mathematical model. I want to introduce you to the mathematical model. Actually, it is a very easy one. So we have two types of ecDNA, blue and red. I call them with two different colors just to make it easy to distinguish them. And blue ecDNA and red ecDNA have a reproduction rate each. So the one of blue is SB, the one of red is SR, and these reproduction rates are parameters that are greater or equal than 1. Then we can consider two probabilities that are called PB and PR that are basically modeling the interaction between the two species. So at each cell division, with probability PB, it is possible for blue ecDNA to turn red, and with probability PR, is it possible for red ecDNA to turn blue. Basically, if we let the population grow from, let's say, a single cell with a single copy of blue ecDNA, we could have as many different scenarios as possible. And also we can distinguish some subpopulations that are based on the number and type of ecDNA copy that we could have. We can indeed distinguish 4 cell subpopulations. The first one is pure blue ecDNA cells that are carrying just the blue ecDNA, then pure red that are just carrying the red, then mixed ecDNA that are carrying both red and blue, then finally cell without ecDNA. Of course, all these compartments are highly related because at each step it is possible for blue ecDNA to turn red, for red ecDNA to turn blue. As I said before, we need to model in a way the random segregation of ecDNA into cells. And we model that by using a binomial distribution. Why? Because it is highly used, especially in population dynamics, for random processes of segregation. Before starting with the results, I also want to introduce you to the measure of entropy that is the most used in biology, and this measure of entropy is called Shannon Diversity Index. Basically, the Shannon Diversity Index is a measure that we use in population dynamics to basically try to understand how diverse and how even the population is. So this is the formula we use for this index, where CI is the abundance of category I. So when we want to calculate the Shannon Diversity Index, what we do is defining categories and then applying the formula. If we want to compare different scenarios between each other, we can also consider the Shannon Equitability Index. That is basically a normalization of the previous index. So we take the index and we divide that by the number of categories. And in this way, we can normalize the Shannon index in any different scenario, and we can compare them. Okay, so let's see some results that I got so far. First, if we consider different processes with different reproduction rates and different switching probabilities, PB and PR, then we can appreciate that some quantities are not depending on PB and PR. Just, I go back for a second, just remember that PB is the probability for blue to turn red, and PR, red to turn blue. So it doesn't matter how these two types of ecDNA are related with each other, some quantity stays the same. For example, if we look at the distribution of copies in cells regardless of subpopulation, then we can appreciate that this quantity is stable for every different value of PR and PB. Same for the density of ecDNA-free cells. So the quantity of cells that are not carrying the mutation, I mean the percentage of cells that are not carrying the mutation among the whole population, this is the same for every value of P when we fix the reproduction rates. Also, we can mathematically measure this fraction of cells that are not carrying the mutation. If we consider reproduction rates equal 1, and like in bioterminology, it is called neutral selection. It means that if we have a cell that is carrying a mutation and a cell that is not carrying a mutation, they are growing with the same reproduction rate. Then in this case, we are expecting the trend of ecDNA-free cells to go to 1. So it is increasing until taking over the whole population. If we have positive selection, so it means that the reproduction rates of blue and red ecDNA are greater than 1. So it means that if a cell is carrying a mutation, it is growing faster than a cell that is not carrying a mutation. So we say positive selection. In this case, the density of ecDNA-free cell is shrinking to 0. And how fast? Well, it is an exponential decay, as you can see. And this exponential decay is related to the reproduction rate through this formula that we found using solving the differential equation and also some fit techniques, like best fit techniques. Now, I want to show you some distribution aspects of our population that is deeply depending on our parameter. If we consider the population starting from a single cell with a single blue copy, and we are considering the same switching probabilities between species, so blue could turn red with probability P, red could turn blue with probability P, then we can appreciate how the population is differently composed using different parameters. Just to explain with these pictures. So here on top, you can see different values of P. So low P, high P, intermediate P. Also, blue means pure blue population, so cells are just carrying blue ecDNA. Red means pure red population, so cells are just carrying red ecDNA. And purple means mixed ecDNA population, so cells that are carrying both mutations. And the different shapes of the line is related to different selection scenarios, so different reproduction rates. And we can see how the population is deeply changing, not only for the different reproduction rate scenarios, but also for different interaction that the cells could have. And we can highly see that if P is intermediate, then it is much visible how the mixed ecDNA population is greater than all the other scenarios. So basically, the subpopulation that is more affected by that change in the interaction between the two ecDNA types is the mixed population. And I will show you later some study that we have done on the mixed population that is also very interesting for us, because of course, it's the population that is carrying at the same time the two types of ecDNA that we want to study. Also, the composition of subpopulation is strongly depending on initial values. Of course, we need to be able to understand the property of each population for every starting point. Like we can start studying the population at 1 cell, we can just start studying the population at 1,000 cells, or etc. And so we need in a certain way to understand how the dynamics is related to the initial value. Unfortunately, we thought at the beginning that our system wasn't depending on the initial values, but unfortunately it is. That is unfortunate in terms of mathematics, because of course we cannot get some very nice results for a system that is strongly depending on initial values. But in terms of therapeutic strategies, it is actually pretty cool, because we can understand how to treat different types of cancer and how to control the mutation, because we can basically have different trajectory for different starting point. Here, for example, we started from a cell with 1 red and 1 blue, and we compare with a cell from a cell with 1 red and 2 blue. Let's look, for example, at the purple trends. The light purple is related to 1 blue, 1 red. The dark purple is related to 2 blue, 1 red. As they are not converging, we can basically see how starting from a population that is more numerous in terms of copies lead to a percentage of cells that are carrying both mutations that is much higher. So basically, this could be not only interesting because there is a complex system of trajectories, but it's also interesting if we want to control the mutation and we want to see in the long run how the mutation is going, compared, of course, to the initial values. I want to also show you some diversity aspects. So we perform a deep analysis of the entropy of the system. starting from Shannon index at whole population level. Here on this table, I just put some explanation for you. So what does it mean that we are considering the Shannon index at whole population level? We are basically taking as categories each configuration of the cell. So if a cell has 2 blue and 1 red, it is part of a configuration. If a cell has 2 red and 1 blue, is another configuration, and so on. So we calculate the frequency of each configuration, and we then apply the formula for the Shannon index. So this process allows us to understand how much diverse the population is on its overall. Also, we normalize these indices for different scenarios in order to compare them. So if we consider reproduction rates equal 1, reproduction rates greater than 1, or different reproduction rates for both blue and red ecDNA, then we can see that no matter P, all the Shannon index are clustering. It means that basically there is a linear dependence between P and the number of categories. So what's going on is that we can say that if P is at intermediate value, I am expecting a higher number of categories, so a higher number of configuration. And this means that, of course, the higher number of configuration is led by the mixed ecDNA population. That was the most numerous in terms of intermediate values of P. This is also well explained if we look at the diversity for each single cell. So if we take a cell and we consider as categories the density of blue ecDNA copies in a cell and the density of red ecDNA copy in a single cell. So basically we are taking a cell and we are calculating its own Shannon index. Then we can plot these results, and we can basically plot a heat map that shows us how frequent each configuration is appearing in the population. And we can see that the darkest the dot for each number of ecDNA copy, and the most frequent the related Shannon index for every cell. So basically, for example, if I have 10 copies and I have this value of the Shannon index, it means that this value is at a certain frequency for cells that are carrying 10 copies, and so on. We can also calculate what the most frequent Shannon index value is, and we found that the most frequent is always this one that is related to cells with 3 ecDNA copies. So basically, for every value of P, for every single reproduction rate scenario, we are expecting that the population is dominated by cells with 3 ecDNA copies. So this is very interesting in terms of validity of our model, because some data have shown that if we have a small cell population, then the majority of the cells is actually carrying 3 ecDNA copies. Finally, you can see that here in the middle, we have the highest diversity. So we have the highest diversity of points rather than lower and higher P scenarios. So there is the higher diversity when the mixed ecDNA population is larger, because it is larger for the intermediate values of P. And also, this is a proof that the number of categories, as I said before, is linearly depending on P. I want to finally show you something new that I'm working on in the past month. This is called ecDNA hubs. So just to explain you what it is. An hub is a cluster of ecDNA elements which aggregate in cell division. Basically, let's say that we have 2 types of ecDNA that are not related with each other. So there is not a dependence, a cooperation between them. So let's say that P, that was the switching, is equal to 0. Then at each cell division, these 2 types independently random segregate. So let's say that here I have 2 blue copies, so I have to distribute 4 copies, and I could do 3 plus 1. And for red, I can do 2 plus 0. With a certain probability lambda, an ecDNA hub could be formed. And an ecDNA hub is basically a cluster of the higher number of blue ecDNA copy to distribute, plus the higher number of red ecDNA copy to distribute. So basically, when an hub is formed, we are expecting that the highest number of ecDNA copy to distribute is linked to the highest number of red ecDNA copy to distribute. And with probability 1 minus lambda, the opposite is happening. This, of course, is interesting if you want to understand the mean copy number of the mutation over the population or the composition of the population itself. Because, of course, introducing the existence of ecDNA hubs could deeply affect, in general, the random segregation. I want to underline that I put here a reference because actually this is a very, very recent result. ecDNA hubs are not present in every tumor. There are some tumors where we could appreciate ecDNA hubs at some stages of the tumor. For some tumor, this doesn't happen. So it is still some work in progress. But of course, it's interesting to see in terms of a mathematical point of view and a distributional point of view what's going on. For example, if you consider a population that is starting from one red and one blue copy, and we are not considering a switching between species, so p is equal 0, and then we are considering lambda as a clustering probability. So if lambda is equal 1, then there is an ecDNA hub in every division. So highest blue plus highest red. If lambda is equal 0, then we have highest blue plus lowest red, because the opposite is happening. And if lambda is intermediate, then we have just a probability of clustering happening or not happening. Here I can show you some simulations. For example, the composition of subpopulations is deeply affected when ecDNA hubs are occurring. For example, here we can look at the ecDNA-free cells. The fraction of ecDNA-free cells is different if lambda is equal 0, 0, 5, or 1. And it is different because, of course, if a cluster is happening, then I am expecting that with a highest or lower probability, also a cell could have no ecDNA at all. And of course, if lambda is equal 1, then I am expecting that as I am clustering the highest copy with the highest copy, then it's much probable that a cell is remaining without any copy at all. So the proportion of cells that is carrying the mutation is not carrying the mutation is expected to be higher at the beginning. Also, if you look at the mean copy number, we can see that the mean copy number is always higher when lambda is equal 1. And this is expected because, of course, if there is an ecDNA hub, then I'm expecting that a lot of cells will have an highest number of copies. Just to summarize, first, our work is biologically motivated. I didn't give the introduction just because I wanted to give you an introduction, but it was also for motivating the work we are doing, because of course, behind this model and behind some assumption we have, there is a lot of literature and a lot of medical research. Also, our model is pretty versatile because we can adapt the model to different real-life proliferations. So, for example, also the study of ecDNA hubs, I'm doing that by taking the original model and do some modification. Also, this is the main goal of our team worldwide. Basically, we are not studying ecDNA just for mathematical purposes or for clinical purposes. We are studying that because in a certain way, our study and our predictions could contribute to better understand the relation between cancer and the chromosome instability episodes. So the occur of this mutation, basically. And finally, so far, the main achievement is that we have related the random ecDNA segregation to peculiar dynamics. So even if the segregation of this mutation is random, we can basically make predictions. So this is good because, of course, we are not leaving this mutation to grow as it wants. Yes, of course, it is doing that, but we can in a certain way predict how a population is going to be from the starting until a certain size. So yeah, so far, thank you for your attention. And I have also something to discuss with you in terms of the problems that I encountered in this project, because unfortunately I had a lot of problems going on in the project, a lot of breakdowns. So I'm more gonna have to do open a discussion. And if you have any question, I'm open to that.
Okay, so I was muted, I couldn't unmute. Thanks a lot, Elisa. Thanks a lot. Let's see if in our wonderful audience we have some questions.
I have a question.
Silvia was expecting it. Go ahead.
I am curious. Thanks for the great talk, Elisa. And I'm curious about the three copies dominance. Do you have any guess, any idea on why that specific number, that specific configuration is the more preferable, if you want? Or...
actually, this is a very good question.
I think it's something related with equilibrium or stuff like that, right?
I think so. I mean, we think so because actually some data have shown that. So there is, I think it is more like maybe a way how the species can cooperate together. Or maybe it is related also to ecDNA hubs in a way that maybe when an hub is occurring, then few copies are left for the other cell, and especially when the population is pretty big, maybe just few cells are left in the other daughter, and maybe these few cells could be around 3. We still don't know though. There are some evidences, and also a study about a single ecDNA cell has been done. So like a single ecDNA type, so no two types. And the people that have done that, they found again that 3 was the preferable number. So we still don't know. There is this going on. We have, of course, to further investigate it. Of course, this is just so far just a distributional, let's say, result. We don't have a biological reason behind that. We have a biological reason for lots of other results, but for this, no, to be honest.
It's really interesting. Also because like a triple configurations is the one, if you want, that is optimizing the energy because you're using the less number of copies. But at the same time, you are optimizing, if you want, the resistance, the robustness of the geometrical configuration, if you want. It's like it's the less, but the most powerful, if you want. So it makes sense that is 3, the number, the dominance number. But very cool, very nice. Thank you, Elisa.
Thank you.
I also had a question about like I don't know if you already had or maybe you're planning to, but like some specific things that you could actually apply and tell the clinicians, okay, do this. Like what could that be, or what is that being already with your work that you presented us?
Yeah, actually, we are waiting for data. So yes,
Okay.
and we have data for ecDNA hubs. So basically, next step, I think, new year, we are applying our prediction to data. So to see if the mean copy number and the composition of subpopulation is matching the data. We already have them. Also, we are, just to say, I go back. Oh, no. Okay, yes. Okay, in this model, basically, the next thing that we are going to do next term is considering PB and PR as functions, not probabilities, and apply the so-called co-selection, because this is much easier to be measured. It means that the cells are cooperating, so these PB and PR are not probability, but are functions that are depending on SB and SR. And in this way, we can collect data, because it means that the cooperation between the cells is that they are not turning one into the other, but they are cooperating in the sense that when they are together, the reproduction rate of the cell is much higher. How higher? It depends on a function. So now this is like the next step we are going to do in our model, in a way that we can collect data and we can compare to data.
Thank you. Let's see if we have other questions. Otherwise, we can move on to the next part, and you can share your experience from there. We see where it goes.
I have a question.
Lisa, go ahead.
Yeah. Thank you very much for the talk. Very interesting. At some point you said like that the result of the model depends on the initial condition. So my question is, how much depends on the initial condition? Because maybe from a therapeutic point of view, you don't have access to the very first point, but you have access just to an early stage of the of cancer. So what I mean of the condition of the patient. So
No, yeah, this is a very interesting question, and it is I'm more than happy to answer, because it is depending on initial value, in the sense that if we start from a size of the population, especially if we start, let's say, the cooperation between the species at a certain size of the population, or for example, if there is a population at a certain point, a new type of DNA appears, let's say that, then in that case, the whole scenario could be predicted, but will be different, instead of having a small population and then the mutation occurs. So it depends, in the sense, if we have an early stage cancer, our goal is like to be able to measure the size of the cancer first, then the composition of the cells, and to put everything in the model to predict that. But we need, that's the point, is that we do need the initial population. We do need the measure on the initial population, because it is not true that if the initial population is small, large, or just composed by certain types of cells, then the result is the same. This is maybe, I don't know if I answer your question, but it's basically deeply depending on the initial value, in the sense that we do need to understand deeply the initial value, because it matters a lot when the two types switching starts to occur, let's say that.
Okay, thank you very much.
So maybe now you can say something. I don't know, you had something planned?
Yeah, okay. So actually, I prepared a slide.
Nice.
Just for remembering it, because
amazing.
Okay, so I want to start with the last one. It's like, okay, I started my PhD in 2020. And as all of you know, I am from Italy. So I came here in London, and it was 16th of December 2020. So it was like tomorrow of 2 years ago. And when I came here, my feeling was like, hey, amazing, I will cooperate with a lot of scientists. I will have a lot to talk about to everyone. But actually, that wasn't this way. So my first year of the PhD was a complete waste of time. It means that I was trying to do my project, and I did something, but then everything was completely no sense, because I hadn't anyone to talk about my project. I couldn't find anyone who can understand what's going on in my personal PhD project, especially because, unfortunately, in London, we are very few people that are doing mathematical biology. And at the time, I didn't know anyone that was doing mathematical biology. So my feeling was like, I was feeling super isolated, but not just in the sense that I was feeling isolated because everyone was isolated in our houses, but I was feeling isolated in terms of my academic career, my academic path. Because, for example, I was talking to some of my friends that were doing PhD abroad, and they were from my university, but no one of them could understand, because they were doing other projects. And I couldn't know anyone that was doing my same project or similar project. And the situation of COVID-19 that basically made us so isolating on our own made me very isolated in terms of my academic path. And I basically wasted an entire year, because my project in the first year was similar to this one, but it was a project that had literally no sense. So it was a bunch of simulation put all together, but the sense was absolutely anything. And now, like, now, of course, I overcame that, because luckily now the situation is much better, and I know a lot of people I'm collaborating with, so nice. But I have a lot of bad feelings about not being able to produce something sensible, in the sense that I'm still in progress on writing my first paper. So I have a draft, is there since months, but it does still like to be published and everything. And every time that, like, I'm looking at that, I'm like, am I writing something sensible? Am I doing something sensible? Or this is just a bunch of nothing? So that was basically the major difficulties I encountered, and I'm still encountering though. Of course, like, the lack of motivation is something that I think everyone encounters at least once in our PhD path. Of course, like, also the imposter syndrome. I think that everyone encountered that in our lives. In my case, that was pretty big, a big issue, and it is still a big issue, in the sense that sometimes I'm feeling Am I doing something that it has sense? Am I finishing my PhD, or am I just like stuck in a project and I'm not able to go ahead? I don't know if you are sharing similar stuff, but like, especially the how can I say, like, collaborating now is not a problem anymore, but like saying, can I produce something that makes sense? That is still something that I am dealing with, like, literally every day. So it's not just something like, oh, okay, yeah, today I'm feeling this way. No, it is something that I do feel a lot, to be honest. And I feel that it's deeply related on the fact that, of course, we are living in a sort of time of history that is pretty difficult. And we came from 2 years of complete isolation that, of course, in our case, because we are all doing PhD or everything, in our case was pretty dramatic, because it affected a lot of our projects. Of course, then there are a lot of different, how do you say, factors, because also, as I said, the fact that almost all my collaborators are in US, this is a problem. Because of course, like, you cannot meet them unless you are taking a flight of 12 hours. And so it's not extremely easy in this way.
Yeah.
Also because the meeting, I think the difference of timing between the time zones is
No,
not.
it's great.
Yeah.
Like, when we're meeting, it is like 7 here and 9 in the morning there. So it's not like, it is not something that you can say, okay, let's meet up tonight. It's not this way.
Yeah. Working with the US is like that.
Yeah.
What do you think when you say, is it my work sensible, makes sense? What do you, what's the work that you think that makes sense, for example?
Okay, like, I have a project and I know that it makes sense, but the problem is, how can I take everything together and say everything together, let's say biology, genetics, mathematics, data, how can it make an overall sense? So sometimes I'm a bit lost in that. So this is something that I am encountering, to be honest.
Yeah, I think it's also because you are working into like a very interdisciplinary project.
Yes.
Because the funny is that I'm also working with biologists, but in another total different field, because I'm working with plants and that kind of biology. So it's totally different from cancer, from if you want humans and stuff, human cells and stuff like that. So I think it's true that also when you work in a, I don't know, network theory field, okay, that is something, or complex, I don't know, complex theory, something more related with theory, that it's something that you can control better if you want, because it's our field. Also in that case, you can ask yourself, I'm making this work is sensible, makes sense or not. But I think that this feeling is also greater when you work with I don't know, biologists, chemistry, engineering, with different fields than yours. So it's something that is also enlarged by the difficulty of like putting all the pieces together, as you mentioned, the maths, the biology, also because we are not biologists, right? So
no,
also the question I ask you, why the 3 copies, and you say to me, I don't know, because I'm not a biology, which is
yeah, I mean, you're right. And also sometimes I think that I'm feeling that I'm not able to produce something sensible, because if I'm presenting my work to someone that is, let's say, a biologist, sometimes they don't understand, because of course they are not mathematicians. Or sometimes, like, I'm listening to their talks and I don't understand because I'm not a biologist.
Yeah.
Also this problem in communication between the different disciplines. And of course, this is something that you cannot overcome so easily. And sometimes these make me feel, okay, maybe I'm not doing something sensible.
Yeah, no, I agree. I also had a project about brain HEG data. No, and it was like during an experiment basically with two people, and so you had to study this hyperbrain interaction between the two, and so for me it was strange. I had a very different, I don't know if it was similar or not to your experience, but basically the things that mattered in my opinion was the fact that they were explaining everything to me, but just the things that I needed to know to understand what I needed to do, and then produce the results, but never really understanding. Okay, like you number 8 has a higher centrality. Okay, but what is, I don't know
Yeah.
what for the brain
Yeah.
is that. And
Yeah.
so then I
happened to
say,
me
then
too.
what happened was like say that to them and to the main the PI basically and the postdoc, and they was this interesting. So then basically the parietal blah blah blah, and so but this is incredible because then they ask, oh, so maybe can you check these things on that and that and that, because it makes sense that that would appear blah blah blah, and this would become very great. But this was good because they were putting the effort. Like I was like, okay, I have the analysis, and the same day or the next day we would meet online because it was COVID time. While opposite of this was like another project about the same things I'm doing for my PhD, while the other one was an external project. And there instead it's impossible to meet. I like, for example, we received the review, we got rejected, and it's been 6 months already.
Wow.
And we met once. I mean, I did some work, but like the PI of the project doesn't know anything. And then after a month, I didn't reply. I sent another email, and maybe we're gonna meet on Monday. But I mean, this of course, that's when I lack motivation as well, because like
Yes.
we're in a similar situation than you in different reasons, but it's very bad. And that, like, you have your project, but you say, oh, but it's been 4 years that it's been going on. And so it feels like you have not done anything, even if it's a nice
that's the
project.
point, actually. I mean, the point is that when you are working, especially with people that are not from your field, and you are one of the few that is a mathematician or a physicist, it is pretty tough to say I'm producing something sensible. Because, like, for example, when we had the big meeting with all the group, we are almost 70 people, and let's say that 70 people, and there are 6 theoreticians. Yeah, we are 70 around the world. And we are like 6, maybe 7 theoreticians. So of course, like, we are talking and people are making like some questions that are like, I don't know, let's say, but why have you used this equation? And of course, like, it's difficult. There is a sort of difficult in communication. There is a sort of difficult in understanding each other, as you said.
It's also a lack of feedback that you are mentioning, right?
Yeah,
Because
of course.
if we don't have someone that says to us, this is bullshit.
Yeah, but in fact,
it's difficult.
well,
It's really hard.
in my case, we were much less. We were like 10, but there were like 3 different groups. Was one group doing the analysis and all the project, another group suggesting methods to do the data analysis, and then us was doing network analysis. But we were only speaking, the 2 groups were only speaking to the main group and the PI, and like I whenever the feedback was only between me and the other 2 guys on Nectar side, we were only speaking between us. And then when we were talking to them, we were saying, okay, I'm not gonna say how this centrality or this measure, whatever, is calculated. I'm just gonna say what it means. And so this was the way we communicated. Like I never showed the equations. I wrote that part of the paper myself.
It's like that.
You know, so you have to understand, the idea behind this is that, you know, it proliferates and you can split, so you have some probabilities, and these results are this, boom, without, and I'm gonna explain you the results without knowing the mathematics behind, because you're not gonna understand. You're gonna get confused more than if I just show you the picture. So that's, I think, the way to go.
No, but I do agree. The problem is that, yeah, sometimes it is just, as you said, it's just affecting your motivation, and it's just affecting in general the way you are feeling your project in a certain way.
Yes, that's true.
Yeah. But yeah, at the end, I'm glad because like I literally started by a project that was wrong since the first page until the last. So at the end, now it's not completely wrong. So this is just an achievement. Then we'll see later.
Nice.
I still 1.5 years to work on it. So
Yeah.
I don't know if anyone else has something to add on this topic.
I was thinking also about the stuff Gabriele mentioned, that having like a very strict deadline, for example, for the
Yeah, they had a deadline.
for the project with the biologists, the cool thing, one of the cool things, because I really love this project, is that we meet like every week. So every week we have on Monday, that is the time, we have to produce results. We have to talk about what we did during the week. So I had to work on the project. So I had a lot of motivation from that. Also because it's pretty finished. We are starting another one with the same group because we work very well together. So the pretty thing was that every week or maybe every 10 days, but never more than 10 days, we have a meeting. So we talk about the project and we move on. So in like a few months, we produced tons of results. That is very nice. When you on the other hand, when you have like this very slow process in have a meeting and talk and share results, also the motivation is totally lost. Yeah, I totally agree. And now we are PhD, so we have to like take quite all the projects that arrive to us, but in the future maybe it's good to give away the lower one, because it's totally a waste of time, I think. But now it's like that.
Now it's gold.
Yeah.
All right.
Can I add something on this line?
1 more minute.
Oh, thanks.
No.
No, that's really an important point, that you do a project that motivates you, and the fact that at the beginning we don't choose a project, basically most of us don't choose the project, can really slow down things. I mean, it happened to me at the first year. I had 2 projects. One motivated me a lot, but it didn't bring to nothing, so I should stop it. And the other one was more or less okay, but didn't motivate me so much. So I was so slow the first year and didn't produce actually like half of the thing, but then we got stuck for like 6-7 months on the same on a just single problem, because it was not my first option. I didn't realize at the beginning that the problem was motivation for me, but it's really important to be motivated on what you do, because it's, I think, the full that gets you through all the problem that you can encounter.
Yeah, no, I do agree. Yeah.
Okay, thanks Elisa, thank you all.
Thank you.
And
Thank
it
you
was
for
a
the
great
discussion.
Yes, it was a great discussion indeed. And so the next talk will be on the 12th, I think, if I'm not mistaken. Should be on the 12th. Yes, 12th of January. And so yeah, we'll announce it very soon.
Yeah,
all right.
I think to join more often. It's very nice.
Yes, you should tell everyone you know about NetPlace.
Bad news, no worries. Thank you.
All right. Thanks. Bye.
Bye.
Bye.